Good evening, all.
Good
Hello.
evening, sir. Yes, sir.
Right.
Good evening, sir.
Good evening. Last class we discussed about the DNA synthesis
DNA
and
replication.
yes, DNA replication or DNA synthesis, both are same. Right.
Yes.
Phytohormones also completed. Right.
Yes, sir.
So, actually, due to the poor signal, we were unable to complete the DNA synthesis. Now we are going to complete DNA synthesis and next enzymes. First, let me complete about DNA replication or DNA synthesis. Already told you the definition.
Okay,
What
sir.
is DNA replication? What is DNA replication? Replication means
Production, the copying of a same DNA
same,
structure, sir.
right? So that is called DNA. Are you able to see this diagram?
Yes, sir.
Yes.
Activities at the replication fork.
Yes. And already explained about the requirement. What are the requirement to the DNA replication? And this DNA replication different in prokaryote and eukaryote. That is another thing. But the common, let me complete with common process with prokaryote and eukaryote. Here are some of the requirement from the DNA replication, such as first one, what are the requirement? One is DNA template, right? First one, template.
Yes, sir.
DNA template. Next, primers. Without primer, actually polymerization activity done by there is newly DNA synthesized strand synthesized by hello.
Yes, sir.
Yes, sir.
DNA strand synthesized by which enzyme?
DNA polymerase.
Yes, sir.
DNA
RNA
polymerase.
polymerase.
No, DNA dependent. DNA polymerase.
Okay.
DNA polymerase. DNA polymerase also required. And apart from that, but this actually the DNA strand always coiling nature, super coiled nature, right? Double helix with
Yes, sir.
winding, winding,
Structure.
rewinding.
Yes, sir.
It should be removed. The winding nature
Yes, sir.
should be removed with help of helicase. Sorry, topoisomerase. Help of topoisomerase. Topo. So this is also another requirement. Topoisomerase. Topoisomerase. Right. Another important one is helicase enzyme, which keeps the DNA. That means it is cleave the hydrogen bonds between the two complementary DNA strand. Helicase. And another one is single stranded binding protein. Single stranded binding protein also required. Single stranded binding protein prevent the rewinding of DNA. Of course, you with help of topoisomerase, you unwind the DNA strand. With help of helicase, you degrade the hydrogens. But what is the nature of DNA? DNA always winding nature. Always winding nature. If you separate it again, rewind. So to prevent this, we can use the single stranded binding proteins. So they are the major requirement for the DNA synthesis or
DNA replication.
Right.
Yes, sir.
Let's see in prokaryote DNA polymerase, there are different type of DNA polymerases involved in prokaryotes. DNA polymerase 1 and DNA polymerase 2, DNA polymerase 3. So different type of enzymes involved. So first of all, DNA polymerase 1. This DNA polymerase 1 activity can synthesize the DNA both directions. That means this activity 5' to 3' strand and also 3 to 5 stand. It can use the template both direction, 5 to 3 and also 3 to 5 direction. Right? For example, this is 5 to 3. What is opposite stand always should be in anti-parallel direction, that is 3 to 5.
Yes, sir.
3 to 5. Right? Or template stand, for example, this is A, this is B. Now the A is the template stand. If the A is template stand, now direction of the synthesis here the 5. So 5 to
Yes, sir. 3
3 direction. So this activity done by polymerase 1. And also same way, if utilize the 3 this template, now where is the template newly synthesized stand 5? Both direction.
500 to 300.
So anyway, so in both stand always 5 to 3, but it can move forward direction and backward direction. That is the importance of DNA polymerase 1. Like that, DNA polymerase 2, only it is synthesized in one direction, 5 to 3. If the template not involved in 3 to 5, it involved only in 5 to 3. That means this is 5 to 3, right? This template stand.
Sir.
Only polymerase 2 activity on this template stand only, but not 3 to 5. That is
on B, sir.
Right.
Yes.
And 3, the plays an important role in DNA replication. It is multimeric enzyme. Actually, most probably DNA polymerase 3 not involved. That means actively involved in DNA synthesis or DNA replication. Right. Apart from that, in eukaryotic DNA polymerases, 5 different DNA polymerases are present. They have been identified in first time in yeast, rat, and liver of human cells. The different type of DNA polymerase alpha, beta, gamma, epsilon. Like that, the different type of. So, but in prokaryote, 1, 2, 3. But in eukaryote, DNA polymerase alpha, symbol different. DNA polymerase alpha and DNA polymerase beta, and DNA polymerase gamma, and epsilon, and another one sigma also. So like this different type of DNA polymerase present in eukaryote. Right. Apart from that, actually forgot another important requirement, that is ligase enzyme. Another one is ligase, opposite action to the helicase enzyme. Ligase enzyme also required. Ligase enzyme also required to DNA synthesis. Let's see the process mechanism, DNA replication. We can see in this diagram. Actually, first, what are the requirement? DNA template. So this is double helix stand. This is first one. This is second one. Both are black color. Both black color stands. So these are the mother stand. So these are considered as a template, right?
Yes, sir.
Okay.
Yes, sir.
Now initially don't focus on blue color stands. Blue color stand, only focus on black color stand. This stand, only
black.
This black color only. Black, because these are the mother stand. Right? First of all, yes, before sir. The replication, if it is prokaryote, only one fork present. If it is eukaryote, number of origin places present in eukaryote. But in prokaryote, as the DNA stand or genome is very less when compared with eukaryote, that's why here only one fork, one origin present in prokaryote. The entire length only one origin. One origin, that the origin means the place of synthesized new DNA stand. Only one present. Whereas in eukaryote, different type of origin fork, different type of origin places are there. In different places, different type of topoisomerase attached, and different places unwind the DNA helix. But in prokaryote, only one place unwind. And after unwind, separate hydrogen bonds, prevent SSB to rewind, again shows continuously newly synthesized strand as a blue color, as it's shown the blue color. Whereas in eukaryote, different multiple synthesis strands are present. Multiple synthesis means, for example, okay, I will explain later. First, let me complete this one. Don't get confused. Okay, right. So in first, actually this is double helix structure. What you see in diagram, double helix structure. In double helix structure, first, the topoisomerase attached here. Topoisomerase. Already told you what is the function of topoisomerase. Topoisomerase function. Hello?
Yes, sir.
What is function of
Yes, sir.
topoisomerase? Function of topoisomerase. Topoisomerase can introduce or remove the turns of DNA double helix. Actually, this is double helix. You can observe here. This is double helix structure, right? Turns into coiling nature. When we bind with topoisomerase, automatically this double helix unwind, right? After unwind, the attached with next time attached with helicase enzyme. What is the function of helicase enzyme? After unwind, now the two strands are opened, right? Actually,
Yes, sir.
before unwind, these two strands are closely, compactly super coiled nature. If it is compactly super coiled nature, is it possible to synthesize new DNA strand?
No, sir.
When it is opened, now it is free.
Then only it can.
Right. Now,
Yes.
now it is free to hydrogen bonds. To separate, that means to synthesize the new DNA strand, first of all, this mother DNA strand, both are separated. This can be separated with help of helicase enzyme. After recruit this helicase enzyme, the helicase enzyme between the hydrogen, between the DNA strand, hydrogen bonds removed one by one. Now completely these are opened. The two DNA, this 5 end opened and also 3 end also opened. Now in next time after that the primers, DNA, these primers, RNA primers are attached at the template strand. These primers can attach one end, 3 end direction, one end. So, always these primers, no. Always these primers attached at 3 end only. Remember it. Always 3 end only. Right? After the primers attached, then only recruit the DNA polymerases. Now DNA polymerases attach where exactly primers attach. After attachment of DNA polymerase, now it is synthesized, shows the polymerization reaction. Now newly DNA strand synthesized, blue color. We can observe this blue color. This one. This blue color strand newly synthesized. Here also blue color strand newly synthesized. But here we can observe one is shows continuously where the 3 end direction, opposite shows continuation direction, 5 end to 3 end. But in 3 end template, but it shows the breakage. This breaks called as Okazaki fragment. Right? Let's just assume one is, let me just wait.
Sir, what is the function of DNA polymerase, sir?
Polymerization reaction, that is DNA polymerase function, synthesize new DNA, added the complementary nucleotide to the template DNA. That is the function of DNA polymerase.
Okay, sir. Okay, sir.
Right. This is 5 end, 3 end. Opposite 3 end, 5 end. Right? Let me change color. So two DNA strands, one is blue color, one is red color. The blue color 3 and 5 end, right? Hello?
Yes, sir.
And red color 5 and
Yes,
3
sir.
end. Right. For example, now here the LK is present. Here LK is attached. Now this activity of LK is now both strands are separated. That means this helix separated. Here one coiled, two coiled. Our convenience had take only three coils. 1, 2, 3. After LK's activity, one is removed. Right? One is
Yes,
removed
sir.
means two remain. Second and third. Right? This is 5 end.
Yes, sir.
This is 3 end.
3 end.
Already told you the primer always bind at 3 end. Right?
Yes,
Hello?
sir.
Right.
Yes,
This
sir.
is the 3 end, right? Now let me change color. Here yellow color primer attached. Now what about opposite? Where the 3 end opposite direction?
On the next stand, sir.
Here this end.
Red
This should be the 3 end because here this is 5 end. Almost this next end is the 3 end.
The next, yes, sir.
Right. Now after that attachment of DNA polymerases.
DNA polymer.
Let me draw D. So here also D. Already told you the activity of DNA polymerase always which direction?
3 end, sir.
Which end to which end? Newly
DNA
synthesized stand.
polymer.
Newly synthesized stand direction. Newly synthesized stand direction. Remember always new stand 5 end to 3 end. So this is polymerase. This is 3 end. What is opposite? The ear. Green color. This 5 end to 3 end. Okay. 5 end to 3 end. What about another stand? Where the 3 end? Here 3 end. Okay.
On the blue color.
Now yes, here the opposite direction. It can lead opposite direction like this. That means already here break. If it is turns into another, if it is unwind another turn, now next end will
then we
begin
will
from
have
this
break.
fork. This fork only. Now this is also another patch. If it is unwind another fork and another DNA also formed like this. For every patch, every turn, one another end, always the DNA stands breakage like this. But what about another? Another shows continuation.
Continuous.
Another stand shows continuation. This continuation stand called as leading stand. This is called as leading stand. But another one is as it goes opposite direction, it always break into fragments. So for example, here the one breakage. So this is one fork. Second here, this is the second coil, unwind nature. So like this, the number of fragments, breakage fragments are formed. This numbers are called as a Okazaki fragment. These fragments, these small DNA fragments called as Okazaki fragments. Anyway, after formation of Okazaki fragment, this should be joined each other. So this activity carried out by ligase enzyme. Now the later this ligase enzyme, they fill the gaps and both the strands join together. Right? After that, both strands are completely formed. Now this is black color and the blue color together form a new DNA strand. The same way here, the blue color and black color together form a new DNA strand. Like this, two completed DNA strands are formed. Okay. This is called replication. Already told you this replication carried out inside the interface of S substage. Right? Why this is mainly occurs inside the cell? Hello? Hello?
Yes, sir.
Yes, sir.
Yes, sir.
Why this occurred inside the cell? Because this sharing the
As
genetic information from one cell to the another cell.
okay, sir.
Okay, sir.
Just wait, let me clarify. This is the one parental cell DNA, double helix DNA in the form of chromosome. In A space, what happened? To DNA replication in A space, DNA replication. This is one DNA. This is one DNA. So each DNA strand can prepare newly blue color. Newly DNA strand. Right? That means copy the information. During cell division, that means this is G1, for example, in S phase form. This S phase DNA replication takes place. As a result, these two strands become 4, right? Hello?
Yes, sir.
Right. After G2, all the arrangement are formed in G2. Later, division phase. What happened in division
Yes,
phase?
sir.
In division phase, this cell divide into two
Daughter
daughter
nucleus.
cells. In
Daughter
two, after divided, two daughter cells always here. Right. What happens? Stably maintain. Each cells the same genetic material stably maintain. To maintain stability, first of all, it should be multiply. For example, this is the daughter cell. In daughter cell, how many DNA strand present? Hello?
Yes, sir.
How many, for example, here, how many
Yes, sir.
DNA strands?
2.
So, okay, how many chromosomes are present here? Only one chromosome with here double helix DNA, here double helix DNA, right? In each double helix DNA, convert into 4. After separate, again formed into same
4
number.
only, sir. Yes.
This is the mother cell.
Okay.
This is the daughter cell. Any difference between mother and daughter cells in term of
No, sir.
DNA?
No,
In term
sir.
of
No,
DNA?
sir.
No. Right?
Yes, sir.
So that's why this DNA replication takes place inside the organism. Is it clear? Any doubt in DNA
Yes, sir.
replication?
Yes, sir. No, sir.
Yes or no?
No, sir.
Right.
No, sir.
What is next one? After DNA replication, protein synthesis also given, I think. Protein synthesis also there. Already we completed protein synthesis, right? Hello? Another function, the page number, DNA. Unit number 5. Right. So unit number 7. Nucleic acid, unit number 7. DNA structure completed. Watson-Crick model completed. RNA already completed during the protein synthesis. And our DNA replication and transcription. DNA replication, just now we have completed, and transcription already completed, and translation means protein synthesis mechanism also completed, right? Hello?
Yes, sir.
Just wait, transcription, translation completed or not? during genetic code formation in plant physiology. Right, in plant physiology, we already completed nitrogen metabolism. In my nitrogen metabolism 15. Okay. In 15th nitrogen metabolism, sulfuric metabolism. This version we completed biosynthesis of protein, genetic code, transcription, translation. We already completed. Okay. Was it completed or not?
Yes, sir. Yes,
Right.
sir, it is completed.
Yes, sir.
Right. That's why no need to again repeat it. Now next one, enzymes. Enzyme units 2. First unit, that is thermodynamics, I think. What is first unit? Principles of thermodynamics.
Yes, sir.
Yes, sir.
I will get it explained by the chemistry sir later. I will ascend you.
Okay, sir.
Next, enzymes and carbohydrates, lipids. Okay. Protein, nucleic acid completed. Amino acid, proteins remaining. Correct. No problem. Now enzymes.
Sir, excuse me.
Hmm.
Sir, we have submitted the assignments, sir. You have collected it from zoology, sir.
Yes, I collected.
Okay, sir.
Right. So enzymes, what are the enzymes? Can you give general definition of enzymes? Enzymes simply, these are the chemically proteins. Enzymes are chemically proteins, right?
Yes,
And
sir.
let's see the properties of enzymes. Here given the properties of enzyme. Right. Specificity. Enzyme have specificity. Next, high reaction rate. High reaction rate. High reaction rate, or simply say enzyme turnover number. Enzyme turnover number. Specificity, enzyme turnover number, or high reaction rate. See, mild reaction. Opportunity for regulation. Enzyme catalyst. Enzyme catalyst. So these are the major properties of enzyme. First of all, let me complete specificity. High specificity. What is specificity means? Here the enzyme should be specific for specific substrate. One enzyme cannot react on another unrelated substrate. For example, the simple example here, sucrase enzyme. Sucrase is the enzyme. Is it react on protein? Is it react on fats? Hello?
Yes, sir.
Sucrase enzyme react on fat.
No, sir.
Or protein. React on protein. Simple, sucrase is the enzyme Sucrase enzyme should react on the substrate sucrose only. Sucrose. Sucrose. This is the specificity. That is specificity. Proteinase. Proteinase. For protein only. Cellulase. Cellulase. React on cellulose only. So that is the specificity. That this property called as specificity. So the enzyme have the high specificity. Next one, high reaction rate. High reaction rate. Substrate. Enzyme turns into, after reaction turns into product, right? Hello?
Yes, sir.
Yes, sir.
Right. So here the high reaction rate means substrate molecule, for example, 10 substrate molecule react with 10 enzyme molecules, then how much production, productivity, how much productivity formed after the rate of reaction. This number of molecule, this substrate number of molecule, after formation number of product, this number called as a enzyme turnover number. This enzyme turnover number based on reaction rate. This reaction rate, this separate from one enzyme high, for example, here the A enzyme have the high turnover number, B enzyme has the low turnover number, like that. That is the shows the variation among the enzymes. So anyway, how to calculate the turnover number, high reaction, that is calculation, how much substrate molecule involved, how much product formed. That is called enzyme turnover number. High turnover number, I think carbonic anhydrase, right? Carbonic anhydrase enzyme. Anhydrase enzyme have high turnover number, high reaction rate. That means within the time, and also we take the consideration time also. So in enzyme turnover number of substrate, how much number involved in reaction, how much time taken to, for example, here the 10 substrate molecule, 10 enzyme molecule, then the production maybe 50 products are formed. This 50 product within the 1 minute. Within the 1 minute. So this all process called as enzyme turnover number. If you take the carbonic anhydrase, the same reaction, it forms up to 1000 production. That's why it is I value of carbonic anhydrase. Sorry, I value of enzyme turnover number. So that is high reaction or enzyme turnover number. Simple enzyme turnover number within the period of time, how much substrate molecule convert into how much products. So this is this process called as enzyme turnover number, right? Is it clear? Hello?
Okay, sir.
Yes, sir.
Next one, next third property, mild reaction condition.
Mild reaction.
Right. In mild reaction conditions, it is sensitivity to pH and also sensitivity to temperature, low temperature as well as high temperature. Most probably if we take the temperature, mostly these enzymes are very sensitive to the below 10 degrees centigrade, above 50 degrees centigrade. Here in mild reaction, mainly first of all, temperature. If you take temperature, low, inactive, high, also inactive. pH. If you take pH, the optimum temperature, sorry, optimum pH is 5.0. Below the 4, above the 5, enzyme should be inactive state, right? So the simple question, why the refrigerator food materials do not spoil?
Yes, sir.
What?
The temperature which is present in the refrigerator will not allow the bacteria to grow, sir.
Of course, if it is allowed, but the enzymes not active. The enzyme means the bacteria enzyme not active. That means cannot prepare the any toxins. That's why it's take more time to spoil the refrigerator food material. Right? In the same way, in high temperature also. In high temperature also, the microorganisms all are inactive. And pH also, in pH, most optimum pH is 4 to 5 only. Right? But some limitation to the some enzymes, some microorganisms. But in normal, in general, the temperature, optimum temperature, 25 degrees centigrade, 25 to 30 is the optimum. Low temperature inactive, high temperature inactive. That is mild reactions condition. Next one, opportunity for regulation. Here the regulator control regulation, control gene expression, protein turnover number. Again repeated, no need it, ignore it. Opportunity for regulation, ignore it. Next, enzyme as catalyst. Enzyme as catalyst. That means forward direction and backward direction. This is called reversibility. That means some enzyme shows the reversible reaction also. In enzyme catalytic reaction, in enzyme catalyst reaction, for example, substrate plus enzyme substrate complex. Finally, products are formed, right? This is one type of catalytic activity of the enzyme. This is forward. Apart from the forward, it can reverse. Some enzymes can show reverse direction also. That means these complexes again separate into substrate and enzyme. So this is reversibility reaction also present. So this is enzyme as a catalyst. So it can catalyst forward direction and reverse direction. Okay. This is the enzyme properties. Are you clear about it? High specificity,
yes,
high reaction rate or enzyme turnover number, mild reaction, and enzyme as catalyst. Right? So these are the enzyme properties. Now let's see the enzyme structure. Already we know the enzymes apart from the protein part, non-protein part also present. If we take the enzyme, protein part, non-protein part also present. Non-protein part also present, right? This non-protein prosthetic called as prosthetic or cofactor. So now this enzyme called as holoenzyme. Now this enzyme called as, holoenzyme. In holoenzyme, enzyme portion present and protein part present and non-protein part both are present. Now this is called holoenzyme, right? Now another type of enzyme, that is only the enzyme portion without cofactor. Without cofactor, only enzyme. So this combined together called as holoenzyme. But without prosthetic group. Without prosthetic group, this portion only called as apoenzyme. this one called apoenzyme, right? So here the non-prosthetic group or non-protein parts are three types: inorganic, prosthetic, and coenzyme coenzymes. First, inorganic is the best example. Inorganic: zinc, iron, and molybdenum. Prosthetic enzyme group FAD, and FMN, biotin, heme part. These are act as a prosthetic group of holoenzyme. Next, coenzyme. The coenzymes are NADP, NAD, ATP. These are act as a coenzymes. So these are our non-protein parts. These three considered as a non-protein part. Right? Is it clear? Hello?
Yes, sir.
Right, another important one, nomenclature and classification of enzyme. So properties not important in theory, but important in competitive level. And this cofactor, the cofactor of enzymes important as theory as well as in competitive level also. Just in cofactors of enzyme or enzyme structure, enzymes are two types: holoenzyme, apoenzyme. Holoenzyme means apart from the enzyme, non-protein part also present. Apoenzyme means only protein part present. And first of all, what are the non-protein part? Non-protein part may be inorganic, prosthetic, or coenzyme. That's it. Okay. No need to write these reactions here. The given page number 13 reaction also given: donor, oxidized, enzyme reduction. No need to write all this. Okay. Next. Another important nomenclature and classification enzyme. In IUB, according to the IUB, the enzymes are classified. According to the IUB, enzymes are classified, and each enzyme, just wait. Are able to see this? Hello?
Yes, sir.
Yes, sir.
According to IUB, International Union of Biochemistry, enzymes are classified. This should be in 1964, right? 1964. So first of all, the most important one is the enzyme should be the suffix with ase. Proteinase, cellulase, sucrase. Right? Hello?
Yes, sir.
Yes, sir.
Right. But few enzymes are such as pepsin. Exceptional enzyme, pepsin. Renin. These are not end with ase. So these are the exceptional. Anyway, the most of the enzyme, the end with suffix ase. Right? In enzyme classification, they are given here the 4 digits to the enzymes. In 4 digits, for example, one in our book, enzyme code given as a 2711. Another best enzyme code is 111. I will explain later. What is 111? First, it is clear way. Let me explain this one. Enzyme code. So enzyme code should be in 4 digit. Right? In this 4 digit, the first number denotes division, second number subdivision, third number subdivision, and fourth number serial number. So that is the enzyme code. So enzyme code, which category division, which category subdivision, and subdivision, and finally the serial number. So this is common to each and every enzyme, right? For example, here this is the division, oxidoreductase. Oxidoreductase in page number 16. Page number 16. Here the given the table clearly. The first class is oxidoreductase. That is the number 1.
Yes, sir.
Second one is transferase. Hello? Second one is transferases. Third one is hydrolysis. And fourth one is liases, isomerases, and ligases. These are the major division. Right? First of all, this serial number, first one. The first one denote oxidoreductases. Denote the enzyme oxidoreductases. Whereas in according to your book, the name EC code, first number 2. So what is denotes the 2? What is denotes 2?
Subdivision.
Here always the first digit always division, not a subdivision. Please be silent. Hello. Thank you. So first division always, first digit always division. Here the how many
Yes,
divisions
sir.
are given in our book? Here the 6 divisions. Division 1, oxidoreductase. Division 2, transferases.
2 transferases,
3,
hydrolysis,
hydrolysis.
lysases,
Liases 4.
yes,
Isomerases 5.
isomerases,
Ligases 6.
like yes.
So
Yes,
this is
sir.
first division. These are all first division. What you see in this? First division.
Division.
These are first division. That means first digit number denotes digit number. If it is oxidoreductase, number given 1. If it is transferases, given number 2. If it is liases, 4. It is ligases, 6. First number 6. Right?
Yes, sir.
And second one, just see in our book, page number 16.
60.
In oxidoreductase, again subdivision. Subdivision here 1.1. Acting on CHOH
Acting on
hydroxy group. 1.2.
Yes, sir.
These are the subdivision. 1.2, 1.3, 1.4, 1.5, 1.6. The subdivision means here the first one is catalyze the enzyme reaction, whether it is reduction division or oxidation division, whether it is transfer of hydrogen or receive of hydrogen, whether it is release of oxygen or gaining of oxygen. Based on this mechanism, it again divided into subdivisions, right? For example, the first division, oxidoreductase. In oxidoreductase, again subdivided into dehydrogenases and reductases and oxidases. Not given here, but remember this oxidoreductase, first of all, first oxidoreductase, right? In oxidoreductase, first division, subdivision, sorry, dehydrogenase. Dehydrogenase. Dehydrogenase or reductase or oxidase. This is the subdivision. And subdivision means here for example, in dehydrogenase. In dehydrogenase, what is the reaction? What is the type of reaction? Dehydrogenase. In the name given, already we
Removal of
discussed in
hydrogen.
the plant physiology. Right, removal of hydrogen. So this removal of hydrogen from the substrate. From the substrate, removal of hydrogen. So that is the subdivision activity. And what about reductase? First of all, this is removal of hydrogen. Removal of hydrogen. What about reductase? Reductase. In reductase, hydrogen bonds received by substrate. Substrate. One enzyme remove the hydrogen from the substrate. Reductase enzyme received the hydrogen to the substrate. Oxidase. And here the hydrogen transferred to the oxygen. Oxidase removed hydrogen, transfer to the oxygen, as a result formation of water molecule. So these are the different type of activities. These are all oxidoreductase. Division same. Hello?
Yes, sir.
Division same. What is the division here? Oxidoreductase. That is the division. But in subdivision,
Yes,
here, removal of hydrogen from the substrate, adding of hydrogen to the substrate, and removal of hydrogen and adding to the oxygen. So these are the three different type of activities. So that is the subdivisions. And next one, a serial number. For example, here what we given in our book, 2711. According to you, 2711. So to always already told you this division is second number.
Transferase.
Now turn on next page. Internal textured transferases, subdivision 1, subdivision 2, subdivision 3, 4, and 7. 7, what happened here? Phosphate group. In 7, what are transfer? Here the transfer number of transferases. Carbon transferase, kinetic group transferase, acyl group transferase, glycol transferase, phosphate group transferase. If it is phosphate group transferase, the subdivision should be 7, number 7. So this is subdivision. Here the what is the enzyme? Enzyme is glucose phosphotransferase, glucose phosphotransferase or exokinase, right? So already given transferase. That's why it is transferases. That's why we have given the first number, first digit 2.
Yes, sir.
Second one, here the what transferred? Which group transferred? Which
Phosphate
group transferred
group,
here? Phosphate group transferred. Here the phosphate group transferred. That's why the number given 7, because already given in our book the phosphate group 7. Phosphate group 7. If this phosphate group, the transferring of phosphate group to the ATP or substrate or any other material, the based on that again given the number subdivision. If it is ATP involved, number 1, or any other substrate, that is the based on the name, the given the serial number also. So mainly here the involved the ATP. That's why the subdivision 1, subdivision 1, as well as this is the first reaction. That means in transferases, the transfer of the phosphate group in serial number. This is the first one, phosphate transferase. That's why the name given as a 2711. Example here, the phosphate transferase exokinase. Just imagine already we completed exokinase enzyme in glycolysis. Where the exokinase enzyme involved? Exokinase enzyme involved glucose to glucose 6-phosphate, right?
Yes,
glucose
sir.
6-phosphate. Right. Here transfer phosphate group from ATP, not substrate, from ATP, phosphate transferred. As a formation of glucose 6-phosphate. Glucose 6-phosphate. Here the phosphate transferase from ATP is the first one, subdivision first, as well serial number also first. That's why the name, that's why the digit given as a 2711. The same phosphate transferase without involving ATP, if it is this phosphate transferase from the substrate. For example, the same in this same glycolysis, fructose, first last, what is substrate level phosphorylation? Give me one example. Pyruvic acid, pyruvate, right? Phosphoenol pyruvic acid, right? Same way, phosphoenol pyruvic acid convert into pyruvate. What happened here? From ATP, phosphate group added to substrate, or from substrate to from the substrate, phosphorus group removed. Phosphoenol pyruvate to pyruvate. Here also same transferase of phosphate. Okay. Hello.
Yes, sir.
Here also phosphate group transfer. Here also involved transferases enzyme. Serial number okay. And second, phosphate removal, that is 7 also okay. But remove not ATP, but from substrate. Remove from substrate. That's why the changing the subdivision. And as well serial number also changed. So this is enzyme code. So enzyme code 4 division, first two no doubt, right? Hello.
Yes, sir.
First two digit. Subdivision,
Yes, sir.
division and subdivision, no doubt. But slightly confused with about
Subdivision and serial number.
And serial number.
And serial number.
Okay. Don't get confused. Just that is based on reaction. Remember it. Only based on reaction. Okay. Actually, there are number of not given complete structure here also. In our book also not given complete structure. Here up to given only subdivision.
Only they have given subdivision. Yes, sir.
Subdivision. Not given subdivision. But that is just imagine just based on the reaction type, subdivision, and based on number. That means the subdivision may be first, maybe second, maybe third or fourth. That is the based on the serial number also. We will have to mention it. Clear?
Yes, sir. So the division is up to only 6th number. It will not go beyond 6.
Yes, that's it. Only 6 divisions. Any enzyme only 6 division only. Whether it is number 1 or number 2, number 3, number 4, number 5, number 6. That's it.
Okay,
but among this subdivisions, number of
subdivision. Yes, sir. It will vary, sir. According to the division, it will vary.
Yes, and subdivision also.
Okay.
Various in subdivision also. Yes. Right.
Okay, sir.
Just remember only the 6 classes first of all. Okay.
Okay, sir.
And reaction type, just blindly follow this table number 2.3, IUB classification. Oxidative reductase, transferases, hydrolysases, ligases, and isomerases, ligases. And also each one, what they given the subdivision, you will have to mention all this. And type of reaction actually, no need. Type of reaction, no need.
Okay, sir.
First two column enough.
Enzyme.
Yes, first two column enough. Okay. Right. If you possible, just give the example for each class. Oxidative reductase, dehydrogenase oxidase, one enzyme. A transaminase, lipase, decarboxylase, isomerase, synthetase. If better to give one example for each class. Okay.
Okay,
right?
Sir.
Okay, right. Next one, catalyst and enzyme activation, enzyme energy, mechanism of actions. Michaelis-Menten. Very simple way. Substrate product okay, reverse direction okay. Catalysis enzymes of activation. Simple. First of all, enzyme, substrate. When less substrate concentration, less enzyme concentration. Now this both are gradually increased. When you both the substrate and enzyme gradually increase, automatically rate of reaction also, sorry. Rate of reaction also increased. But up to some stage. Now this is first step. In second step, the both are medium. Substrate medium, enzyme also medium. When it is reached medium, automatically try to drop slowly like this. High concentration of substrate. Next, in high concentrate substrate, when we utilize high concentrate substrate, automatically stable the reaction. There is no improvement. There is no improvement. So this is the enzyme substrate reaction. That means in first step, the normal content, less content of substrate enzyme. Now the both are this gradual increased. Substrate molecule increased and enzyme capacity increased. Automatically rate of reaction rapidly increased. But when add and the normal condition, the extra substrate molecule, now but not rapidly increased, the slowly increased, not rapid. In first step rapidly increased, in green line slowly increased. In third step, what happened? This second step. In third step, what happened? You rapidly increase substrate molecule, then the enzyme. In this situation, automatically it reaches stability. That is the maximum. This reaction called as maximum, Vmax. This called as Vmax. So that's one here. The mainly the enzyme reaction activity act to only less substrate molecule, less enzyme molecules. When it's medium concentration, normal growth. When it is high rate of substrate high, the substrate 100 molecules of substrate. But here enzyme molecule only 10. How can these 10 enzyme molecules can convert the substrate into the product? Is it enough to convert the product? Hello? This 10 molecule of enzyme.
No, sir.
That's why when the concentration high, now just concentrate only 10 molecules enzyme. This enzyme 10. First of all, the 8 substrate molecule. If it is 8 substrate molecule, automatically the enzyme rate of reaction increased. When it is 10, almost both are equal, so slowly increased. Now increase all of a sudden 100. It cannot react within the only this 10, not enough to react on the 100 substrate molecule. That's why the fall down. Now this rate called as Vmax, maximum. That is called as maximum rate. Right? Based on this, Michaelis Menten mechanism of reaction I will explain later. Michaelis Menten kinetics. Michaelis Menten kinetics reaction. It reveals Michaelis Menten kinetic reaction. What we in this reaction, first of all, right. So Michaelis Menten reaction, substrate, enzyme, when both are due to the reaction of the substrate enzyme, product formed.
Yes, sir.
This rate of reaction denote with minus dC by dt. Here the C means concentration, substrate concentration. Substrate concentration. Why it is substrate concentration minus dC? Because here the substrate concentration low, minus dC. And D time value. how much time taken, that is D2. So the low substrate, within the something time, the substrate convert into product, which reaction based on substrate concentration as well as enzyme concentration. Let's see in next slide. One, two. Right. As I said earlier, when low concentration substrate, now the substrate low. What is the rate of velocity? Gradual increase, right? Red line. Gradual increased. That's why the
Yes,
concentrate is equal to dC by dt. There is, because first no minus, because when you when you gradually increase the substrate, automatically rate of reaction also increased, but certain time. But when you increased more, automatically the rate of reaction decreased. Low, okay. When the substrate level gradually increased, it is gradually fall down. Later, not a fall down, actually the maintain very slow. In first, rapid improvement like this, and later this improvement very low, and finally it reach maximum. Maximum means no growth of the rate of reaction, right? So minus dC dt directly proportional to C. That means if you improve the concentrate, improve the substrate concentrate, and improve the time,
the rate of reaction will also
rate of reaction also improve.
Yes, sir.
Right? Next. Right, this second one. Medium concentration. Medium concentration of substrate. Concentration dC dt. That means there is no diversely proportional. There is no diversely proportional, because if the concentrate increase, automatically dC dt also increased in the first, like a first reaction, like a first stage. This is first stage. In first stage, what happened? dC by dt directly proportional to C. But in second step, no improvement. Rate of reaction, no improvement. Here, look, this is try to curvature. Just compare the two diagram. This is the rapid increasement. Later, down. This is the second one. That is the medium concentration. And third one, already told you in the third one, one, two, three. Right. Vmax in high concentration. First improvement, second medium, now third fall down. Now it reached the maximum. So minus dC by dt is equal to Vmax. That is third step. What is first step? Minus dC by dt directly proportional to C, concentrate. In second, no variations. That means not rapidly fall down, not rapidly increased. No variation. So this is the first reaction, first stage, directly proportional to last stage Vmax. That is the maximum. Minus dC by dt is called maximum. In reaction, next one. Where it is 1, 2, 3, 4. Right. In first order, low concentrated substrate, V is equal to V maximum into C. Vm means velocity maximum. Km plus C. Km means Michaelis constant. K means Michaelis constant. Now here Km plus C. Right. In first stage, the concentration of substrate low or high. Hello?
Oh.
When compared with Michaelis constant, the concentration of substrate very low, right?
Yes, sir.
Second stage medium, third stage high, right?
High.
So here the low concentrate. The low concentrate, that's why ignore the C. Ignore the concentrate. Now let's draw like this. In second, convert into V. Velocity is equal to V maximum into C. Km plus C. I did not take the C because ignore it. As it low concentrate, ignore the C. Right? Or it can write into V is equal to K into C. That means here K into C means velocity increased as concentrate increased. Is it clear? V means velocity. Increase into means rapidly increased where the concentrate increased. Right? Is it clear? Hello?
Yes, sir.
So this is the first reaction. And second reaction, medium. In medium mixed order, the same unchanged. V is equal to Vm into C, Km plus C. Right? Unchanged. If it is first reaction, C ignore. Why does C ignore? Because the concentration very low. That's why C ignore in first reaction. But in medium, already told you both are normal. Km value as equal as concentrate. That's why it is unchangeable the reaction, this reaction. In third, what happened? When high concentration of substrate, right, high concentrated substrate, already told you V is equal to V max, maximum. Where V is equal to same direction, same equation, Vm into C, Km, already told you Km, Michaelis constant plus C. Now, but here what happened? C greater than Km, constant value. Michaelis constant value greater than C. That means the Km plus C. Km, Michaelis constant plus C is equal to C. What happened here? Vm common into C common. Just wait. Km plus C, C. Right. Km plus C. Right. So this implement here Km plus C. Can it write as a C?
Yes, sir.
Right. So that's why you're
Km plus C is equal to
C. Now both C, C cancel. Now what happened?
So V is equal to Vm.
V maximum.
V max.
Maximum. So that is this is another name called a zero order. Why it is called zero? No improvement. That's why it's called as zero. That is maximum. That is zero order means that is the maximum. So V is equal to Vm. Now finally, the equation is what is Km? In both all equation, Km, right, V is equal to Vm into C, Km plus C. Concentration equal to Michaelis constant. Right? Now what happened? C is equal to Km. So here the Km, Michaelis constant equal to C. C plus C, 2C. Simple here. V is equal to Vm into C by Km plus C. Right? Here, yes, C is equal to Km. That's why this Km place replaced by C. Okay. Now V is equal to, now tell me Vm into C by Km plus
2C.
2C. C plus C.
C.
Right. Now it is converted into Vm into C by 2C.
By 2C.
Right?
Yes, sir.
Now the C cancel. V is equal to V maximum by 2. After this 3 equation, what they given? Velocity. That means the rate of reaction velocity half of the maximum.
Velocity
half
max.
of the V max. Half of the V max. Yes. So that is the final step. The somewhat different with our book. Just follow this reaction. It is the way easy steps. But the somewhat confusion in this. So anyway, if it is not understood, I will get it explained later with the chemistry sir. Okay. If it is clear, no problem. If it is not clear, I will get it explained later. Okay. Right.
Thank you, sir.
Yes, sir.
And next one, enzyme concentration factors, enzyme concentration, substrate concentration. Already these are all repeated. Let's discuss generally. I'm just asking simple question to you. Here the first page number 22, enzyme concentration Vmax, velocity. If enzyme concentration increased, what about the velocity Increased or decreased? See the page number 22.
Increased, sir.
Increased.
It is also increasing.
Right. What about here the second diagram, substrate? If the
Decreasing, sir.
substrate level
Substrate
increased,
concentration, the rate
what
of the
about
reaction
velocity?
or velocity decreases, sir.
No, first how it is the graph? First decrease or increased?
First increase, sir.
Increase, sir.
First increase, later some curvature. The curvature means it is medium. Second stage, already told you, second stage. What is the final stage? Reach the maximum. Vmax, no further growth. Okay?
Then fall down, sir.
Right. Here, the what about temperature? Already told you the optimum temperature for enzymes. Here, the velocity one end and temperature another end. Here, the temperature gradually increase from 10 to up to 30 to up to 35. Okay. 35 gradual increase. What about after 40 degrees centigrade? After 40 degrees centigrade, enzyme reaction goes up or goes down?
Goes down,
Goes
sir.
down, sir.
Goes down gradually. So where is the optimum temperature? 35 degree. Maximum 30 to 40 is the optimum temperature. Right?
Okay.
Just follow the
Okay,
graphs.
sir.
And pH was same pH also. Here the pH, the optimum temperature 7. Below the 7, after the 7, the reaction center completely fall down.
Yes, sir.
Okay, sir.
Clear?
Yes, sir.
Yes, sir.
Right. Let's discuss later enzyme regulation
Okay, sir.
distribution activity. Any doubts?
No, sir.
Are you clear about phytohormones? Last class?
Yes,
Some
sir.
disturbance there due to poor network. Any doubt in phytohormones in last class? Any doubt in DNA replication?
No, sir.
Enzyme?
No, sir.
Enzyme class, properties, mechanism, structure, nomenclature.
No, sir.
Clear so far?
Yes, sir.
Okay, right.
Sir,
Tell me.
hello. Sir, about the previous year question, can you give us class?
Or chemistry? Chemistry class?
No, sir. Sir, about the course, sir, course 4 ka. About the course 4, sir.
This is the course for what you want.
No, sir. About the previous paper, sir. Previous question paper.
Previous question paper, I will give you later. Let me complete the syllabus first.
Okay, sir.
During practice
Okay, sir.
classes, I will give you.
Okay, sir.
Sir, when it will be we are having practicals, sir? In which month, sir?
Maybe
Most
next
probably.
month. Maybe next month. Next month last or May first week.
Okay, sir. The
Right.
theory exams, sir, then?
July or August.
Okay, sir.
Right.
Done.